the Deutsche Bahn arrived.
Yes. Yeah, thank you. Yeah, just arrived from Deutsche Bahn. They had some different plans for me today, so sorry for being late. I'm here today to also talk to you about EPAN, our small little project on plant pan-genomes that I was fortunate to be a part of. This is mainly run by the Belgium node, by Klaas van der Poel and Mieke, and they have put together a team based also on the German node, the Portuguese node, the Slovenian node, and the UK node. And we have set out to look at a few things that we saw that were hindering some of our research in plant science. In particular, lack of standardization and quality control of plant pan-genomes, data integration of different omics layers, difficulty to interpret also some kind of visualization if you look at graph pan-genomes, for example. And we wanted to look at how we could maybe also scale graph-based solution. Looking at some of the examples that we looked at were very much the inconsistent practices that limit repeatability and cross-study comparisons. And one of the main forces there that we saw that in regards to meta studies that we would like to compare to, so was the naming of sequences was very different between different cultivars of the same genome or the same species. Features were named differently. Also, the assemblies had different identifiers throughout. So it was very difficult to find any kind of common ground. Different interpretation also of quality for pan-genomes if it comes to something like the N50 for sequences, or also the protein sequence length, the number of sequences, and so on. Different file formats and so on, but I don't want to bore you because we have already published these results in a small white paper, which you can see here on F1000 Research, headed by the German node, by Mark Heuermann and the other part of the people from the IPK, where we also talk about what I just mentioned, the accession naming, how we should see that this could be implemented throughout plant sciences that would help really in harmonizing data sets. We should adhere to the MyAP standard to ensure traceability of the germplasm. Gene identifiers should use the Darwin Tree of Life system. Assembly identifiers likewise should also follow the Darwin Tree of Life system. And looking at more generalized feature identification of repeat elements, non-coding RNA, regulatory elements, and so forth, where we also propose to extend the Darwin Tree of Life system, which we quite like, to be honest. For quality control, we have also set out a few examples how we see this, how we would like to have it, investigating the quality of the reads that make up the genome in the end, assess the individual assemblies on contingency, completeness, and consensus, backmapping of reads to identify errors, investigate also the annotation of gene and non-coding elements using tools such as BIOSCO, Mercator, Sauron, OMAC, LAI, and TIDK, and then evaluate the pan-genome as a whole, unique genes versus genomes, structural variants and presence-absence variations, gene family expansion contractions. There's also a few slides on data format sharing, and you can have a look after this talk to get a better look of everything in there. And I just would like to mention one thing, is the EPAN workshop, where we also present our results as well as some tools that we are recommending to use on the 18th of May, which will happen online. You can get in touch with me or with other people from the plant community that will be there. And with this, I'm at the end of my small presentation. Thank you.